Tonight's special guest speaker, Dr. Larry Brand, is a professor at the University of Miami's Rosenstiel School of Marine, Atmospheric, and Earth Science. Dr. Brand holds a PhD from Woods Hole Oceanographic Institution and Massachusetts Institute of Technology, and he's a world-renowned expert on toxic algae blooms and their effects on human health. Dr. Brand spent the first half of his career studying kind of the basics of algae, algal biology, physiology, ecology, and evolution. But as more and more algae issues emerged in Florida, he switched his research focus specifically to harmful algal blooms. Tonight, Dr. Brand will explore the different types of harmful algae blooms that are affecting Florida's waterways, their causes, the toxins they produce, and most importantly, the risks they present to our health. Please help me welcome to the stage Dr. Larry Brand.
Okay, well, we're trying to feed 8 billion people now in this world. That takes a lot of plants, takes a lot of fertilizer, okay? It can be mostly common sense, but we're going to start out with a little bit of science here. The two major nutrients you need to grow plants, whether it's algae or higher plants, are nitrogen and phosphorus. And you need them in about a 16-to-1 ratio, about 16 nitrogens to every 1 phosphorus. I want you to remember that number to become important. Okay, well, when you fertilize our crops, okay, not all of it goes into the plants for various reasons. And so some of that runs off into your local waters, down rivers, eventually into your coastal waters, okay? And these are excess nutrients that then can lead to algal blooms, okay? To the extent that those nutrients do go into the plants, humans eat it, but those nutrients don't disappear, they end up in the sewage, okay? So if you just look around the world, you find that most of your large harmful algal blooms tend to be downstream of your large agricultural areas and your large urban areas, okay? But that's not always the case. There's some other things going on, which I'm going to explain a little bit later on, okay? Again, normally it would be in a 16-to-1 ratio, but for certain reasons, that ratio changes, okay? So in some places here in Florida, you'll see 32-to-1 ratio. The plants take it up in a 16-to-1 ratio. What happens then is you take up the 1 phosphorus, the plants have taken up 16 nitrogens, you're left with 16 nitrogens left, okay? This leads to what we call a phosphorus-limited ecosystem. It's a very simple concept, but you're amazed how many politicians and policymakers and so on get this confused and exactly backwards. For example, over Miami, Biscayne Bay, you have these type of ratios, okay? And so politicians say, oh, look at that, all that nitrogen in Biscayne Bay. We don't see phosphorus. There's all that nitrogen. Nitrogen's the problem. No, it's actually phosphorus is the problem, because if you add more nitrogen, you're not going to get more algae because they're limited by phosphorus. If you add more phosphorus, that's going to lead to more algae, okay? And you can have the reverse. If you start out with 16 to 2 ratio, algae take it up in 16-to-1 ratio, you're left with 0 nitrogen and leftover phosphorus. That's a nitrogen-limited ecosystem, and you're going to see both types here in South Florida, okay? Now, major sources of nutrients, okay, again, I already mentioned agriculture, of course, residential fertilizer, golf courses, and of course, sewage and animal waste. These are major contributors around the world. And these are all linked to biology, and so they tend to be in a 16-to-1 ratio, okay? But there's other things that can lead to nutrients as well: soil erosion, peat degradation, which I'll talk about later on, and phosphate mining, and those have very different nitrogen to phosphorus ratios, okay? Okay, other things that contribute to harmful algal blooms is reduced assimilation. Natural ecosystems will naturally suck up a lot of these nutrients, particularly wetlands are good at this. So if you have a mature terrestrial ecosystem, very little nutrient runoff occurs. But if you disturb that, okay, then things are out of whack and you get more nutrient runoff. Okay. Also, if you pave over your natural ecosystems and cover it with concrete, asphalt, or whatever, now none of the nutrients are going to get taken up by the biota or get stored in the soil, and they're just going to run off into your local waters, okay? Loss of wetlands is a big one. Wetlands naturally store up nutrients. They act like a sponge. They suck up nutrients. They're a nutrient sink, okay? We've destroyed a lot of wetlands around the world, okay? And loss of seagrasses in coastal waters, same thing. Seagrass meadows tend to take up a lot of those nutrients, so you're reducing your harmful algal blooms, okay? Once you have a harmful algal bloom, some of the effects of this, some of the ecological effects is it totally alters the food chain, so the whole ecosystem is altered. More algae in the water, that cuts off the light, so reduce water clarity and light getting to the bottom. This leads to the death of seagrasses and corals, which need light on the bottom. So that's a major source. Harmful algal blooms is a major source of death of corals and seagrasses around the world. Also produce a huge amount of biomass. That biomass dies, it uses up the oxygen, and you end up with what's called dead zones. Okay, those are all ecological effects. I'm not going to go into that tonight. But the other thing is many of these produce toxins. I will talk about that in the last part of my talk here, okay? What really got me going on this was in the 1980s, around 1981, a large algal bloom developed in Florida Bay. Now this is Everglades National Park up here. Okay, here's the Florida Keys down here. Okay. My color scheme is red is high abundance of algae, and blue is really low abundance. So naturally, Florida Bay would be all blue. Same thing, here's Biscayne Bay up here. Okay, so this was really unusual because Miami, almost 3 million people, live right there next to Biscayne Bay, and yet low abundance of algae for the most part. Okay, and out here in the middle of nowhere, out in the middle of Everglades National Park, there's no agriculture up in here, almost no people or anything, no sewage. All of a sudden, you develop this huge bloom right there in the north central part of Florida Bay. So the question of what is causing that? No one could figure out what was going on. So I looked into that. Here's just a close-up of it. Oh, and I should point out, this type of algae is called cyanobacteria, also known as blue-green algae. I'll probably use the term cyanobacteria, though, in my talk. Okay? And we'll be talking about that later on, the significance of that. Well, the interesting thing that first caught my eye is the phosphorus in Florida Bay is over in the western part, okay? And the nitrogen is in the eastern part. If it was fertilizer or sewage, things like whatever, nitrogen-phosphorus ratio of 16:1 or something, the two nutrients should be together. So they should be high in the same location. So this tells you there's two different sources. And it's probably not fertilizer or sewage or anything, which we knew it wasn't because there's no sources nearby. Okay? And if you just look at the ratio, the actual ratios here, you don't need to know the numbers or anything, but the red dots indicate a really low nitrogen-phosphorus ratio, so that's a nitrogen-limited ecosystem on the western side of Florida Bay. Go over here to the east, the blue dots indicate it's much higher than the 16:1 ratio, so that's going to be a phosphorus-limited ecosystem. Okay? Well, there's some chemical complexities here which I won't go into, and so I had to check that out, make sure the numbers are actually what's available to the algae. So I do what I call bioassays, nutrient bioassays. Essentially, I'm asking the algae, what are you limited by? And sure enough, the red dots over here indicate they are, in fact, nitrogen limited, and the blue dots over here indicate, yes, they are, in fact, phosphorus limited. Okay? So the basic idea here, let me just see if I got this. Yeah, so the basic idea here is you got phosphorus in the west, nitrogen in the east, and then right in the middle where the two nutrients mix, and roughly at about a 16:1 ratio, that's where you get the bloom. So now we have to figure out what the sources are. Okay, the big puzzle was really the phosphorus, because phosphorus shouldn't be there. And the reason for that is that Florida is really this giant limestone mountain in the middle of the ocean. It's like a giant reef made out of calcium carbonate limestone. And it's well known, and I'm going to the chemistry of this, but basically calcium carbonate limestone sucks up the phosphorus. It'll suck the phosphorus out of the water. And so lots of studies have shown that where you have a carbonated ecosystem, like South Florida, Bahamas, you tend to have a phosphorus-limited ecosystem. Okay? So where's that phosphorus coming from? Okay, well, it turns out, some of you are aware, over there east of Tampa Bay, you've got naturally occurring phosphate deposits for two reasons, geological reasons I won't go into here. And you find them at the surface in these areas where the black is. And there's a huge phosphate mining industry over there. At one time, about 1/4 of all the phosphate fertilizer of the whole world was coming from that area. Okay. Well, and so about 100 years ago, they started mining, first of all, just with pick and shovel. And over time, huge machines are doing it. It's a major operation now. And there's huge areas that have been strip mined for phosphorus. Okay. Here it is in this whole area here. You see those blue rectangles. That's huge pits and so on. And there's mountains of waste material and so on. Here's the Peace River. Nice to see how green it is. So my first hypothesis was you've got all this phosphorus from the phosphate mines coming down the Peace River in the coastal waters and then down south to western Florida Bay. Except, if you look down the lower part, you see the high phosphorus up here coming from the north, but then it gets low, and then it gets high again when you get up to western Florida Bay. Furthermore, if you sample the lagoons in Everglades National Park, which are not connected to the coastal waters, you find that they're high in phosphorus in the west and low in the east. But the fact that you have the phosphorus high in these isolated lagoons, which are not connected to the coastline, and you can't get them from the phosphate mines, tells you there's something else. So I was looking around, I figured, one thing I could figure out something in the geology. Well, it turns out, I saw some unpublished data from a geology colleague of mine, and he had mapped up some of the peculiar deposits he had seen in various well columns and so on. And this turns out those phosphate deposits east of Tampa Bay are at the surface. That's why they mine them. But those deposits continue deeper underground as you move south. So it turns out you have these huge deposits of phosphate under western Florida Bay. They're about 50 feet below western Florida Bay. So that seemed to match pretty good. Okay. The question is, how do you get that phosphorus up into Florida Bay, though? Okay. So anyway, it matches pretty good with the previous geological map. Okay, up here, it's high up here, the 10,000 islands here, and down to western Florida Bay. Well, turns out, I think a lot of people are not aware of this. Remember, Florida's this giant limestone mountain here. Well, as you go down deeper in the earth, of course, it gets hot because of radioactive decay within the core of the earth. And so you just have to go down, and water that seeps into this limestone heats up. When you heat up the water, it rises. Okay, so it's what? Hydrothermal circulation. Okay, and it's slow, of course, but basically what you have is you're shooting warm water up to the surface and then training cold water from either side, from the Straits of Florida, Gulf of Mexico, up to the surface. And we're able to prove this, working with a colleague of mine, Zafar Taub, take advantage, again, don't worry about the details, but take advantage of the fact that uranium decays here to lead, you generate 8 helium atoms, which is a gas. Okay? So if that occurs in rainwater or surface water, what happens to helium? It just goes into the atmosphere. But if it happens underground, it's trapped. It can't get into the atmosphere, so it builds up. So basically, we were able to measure this. Again, don't worry about the details, but all these circles indicate there was excess helium in Florida Bay, indicating that you are getting that groundwater coming up through those phosphate deposits up to the surface. Okay? So that explains, I think, why we see high phosphorus in western Florida Bay. It's actually a naturally occurring thing. Okay? It's been there for millions of years. But that doesn't help us explain algal blooms that occurred only occurred in the 1980s. Okay? So we have to look at the other half of the story. And that's the nitrogen. Where's that nitrogen coming from in the east? Okay, well, if you just look at salinity, purple here is full strength seawater, and the yellow here is lower salinity. That's as a result of fresh water coming out of the Everglades. Water coming out of the Everglades enters in only the eastern part of Florida Bay. So we work our way, here's Florida Bay down here. We're going to work our way back up through the Everglades. Okay, there's Lake Okeechobee up here. In the northern third here, it's called the Everglades agriculture area. That is like 90% sugarcane. That was drained a long time ago for agriculture. Okay? So I'm going to do a transect from the sugarcane fields down to Florida Bay. So here's a transect. Here's the sugarcane fields over here. Here's Florida Bay over here. Okay? So here's your phosphorus. That is fertilizer, and used to provide fertilizer for the sugarcane. But you'll notice it gets sucked up. And again, it's a phosphorus, the Everglades are a phosphorus-limited ecosystem. So all the phosphorus gets used up. So by the time it gets down to Florida Bay, it's all gone. That's why you saw that map, low phosphorus in eastern Florida Bay. Oops. Why? Let's look at the nitrogen. Same thing. Nitrogen's being used up by the plants. But I've arranged the scales here for nitrogen and phosphorus differently, such that if the nitrogen and phosphorus were in that 16-to-1 ratio that the plants need, those two lines would be on top of each other. What this tells you is there's a huge excess of nitrogen. It's more like a N to P ratio of like 100 to 200 to 1. There's a lot more nitrogen than what the plants can use relative to the phosphorus. Well, that doesn't make sense. Why would the farmers be adding a huge excess of nitrogen fertilizer? It just costs them extra money. And what's happening is some of it is being used up by the plants, but once the plants run out of phosphorus, they have no more use for any nitrogen, and the rest of the nitrogen just flows right on in. As you saw high nitrogen in eastern Florida Bay. Okay? But that's not fertilizer. So what's going on? Well, we think of Florida as being flat, but it's not completely flat. Again, for geological reasons, you have a ridge called the Atlantic Coastal Ridge right along the coastline here, and another ridge called the Lake Wales Ridge down the center of the state. And those are what hold the water in between. Instead of water flowing east and west, most of that water tends to be channelized south all the way down from central Florida all the way down to Florida Bay. Okay. So if you look at that from the side, you got a ridge over here, and then a trough in the middle. Okay, well, you start about 5,000 years ago, as we were coming out of the last ice age. Before that, Florida was pretty much of a desert, okay, during the ice age. But now as the climate gets warmer, more moisture and so on, this starts filling up with water. And you develop the wetlands. That's the Everglades. So, and what happens with wetlands is you produce all this plant biomass, the dying plant material falls into the water, it goes anaerobic. And under anaerobic conditions, bacteria can't eat that organic matter, and it turns into peat. So what we have now is like 20 feet of peat filling up this whole trough. Okay? And it sucks up the nitrogen as well. It's stored all the nitrogen in that peat. Well, then humans come along a century ago, and they see the Everglades as a useless swamp, and they want to drain it for agriculture. Well, when you drain that organic peat, you get rid of the water, you expose it to air and oxygen. Now the bacteria can eat that organic matter. Okay? And so what happens over time, you get what's called land subsidence. Okay? You're turning all that organic matter into CO2, which is a greenhouse gas, and you're releasing the nitrogen. So here's an example of it, okay? 1927, that was the surface of the Everglades in this location. Okay? You've lost all that peat in a century. Okay? Including all the nitrogen that was in that organic peat. So what's happened is we have released all that nitrogen that was accumulated over 5,000 years, the history of the Everglades, in the past 100 years. Okay? So that is actually the source of nitrogen. It's not that the farmers are adding nitrogen, it's rather they're taking advantage of this nitrogen-rich soil. They love that soil because they don't have to spend money on fertilizer, or at least the nitrogen fertilizer, okay? So the very fact that you're draining the organic peat is what's releasing all that nitrogen. So that's the source of the nitrogen. Here's another example. This house was built in 1930. It's out in the Everglades. That was the surface of the soil in 1930. And all the soil below the house has disappeared over time. Okay? So every few years, they have to add another step to the stairs here to get up to the front door. So let's look at the history of the sugarcane industry, okay? First half of the 20th century just sort of puttered along. Then all of a sudden it shot up, 1959, 1960, like tenfold. Okay, what's special about that? Well, that's the Cuban Revolution, middle of the Cold War. U.S. government's not happy with a communist dictator only 100 miles to the south. And so they set up this complex series of tariffs and subsidies and so on. I'm not going to go into all that to promote sugarcane growing in the northern third of the Everglades. Okay, but that's in the 60s. But remember, the algal blooms didn't develop until the 1980s. So there's still a mismatch there. Well, the reason is when they decided to drain the Everglades, the Army Corps of Engineers came in and they decided to actually reverse the flow of water in South Florida. They installed huge pumps up here to keep this whole area here dry. They pump all this water back up into Lake Okeechobee and then down the Caloosahatchee River to the west and the St. Lucie Canal to the east. So all that nutrient material, all the waste nutrients are going back up into Lake Okeechobee. Okay? And of course, surprise, surprise, over time, then you start getting algal blooms in Lake Okeechobee, et cetera, and eventually, in a couple of decades in, a lot of lawsuits about this, and so they had to reverse the flow again. But I want to show you data at this point here, which is about halfway from the sugarcane fields in Florida Bay down here. So you see, 60s and 70s, very little flow to the south. This is because all that water is being pumped back up into Lake Okeechobee. Then when the algal blooms developed in Lake Okeechobee, they said, well, we can't do that anymore, so they're going to pump it south. And that's what you see here in the 80s. Okay? All that nutrient-rich water. So you just look at the data here, and you just look at the amount of water now coming into Florida Bay. It starts right there around 1980, 1981. Exactly when you start seeing the algal blooms. Now, we don't have any data on the algae at that time, okay? But the fishermen are out there every day. They see what's going on. They don't describe it in scientific terms, but they can see what's going on. So Kennedy Maria, she interviewed a bunch of these fishermen and said, what did you see back then? And they said, from 1981 onward, the water started getting cloudy. We started having a hard time seeing the bottom. 1981, the water got dirtier, the blooms grew, and seagrass started dying, okay, et cetera. I'll just skip that slide. So just to illustrate this was a drought. Okay, we had because of El Nino or whatever, we had a very dry year. And this blue is high salinity. The pink here is normal salinity water. So the bay got very salty during the drought, not getting any water from the north. Now you come out of the drought, get rain again, and so now what you see is all this fresh water coming in. That's the orange. That's all your fresh water coming in, and you get rid of all that really salty water. Bottom panels here, that's the nitrogen. So during the drought, when there's very little fresh water coming in, you see very little nitrogen coming in. Now, come out of the drought, you start pumping a lot of fresh water into the bay, you start pumping a lot of nitrogen in. And of course, what it does is over here, it mixes with the phosphorus in the west, and you get your blooms. Okay, so what I did is I take data at these 4 points here and look at it over time. Okay. And you see your seasonal cycle, your wet season, dry season cycle. Blue is water flow into Florida Bay in the east, and the green is the size of the algal bloom. So you see pretty good yearly correlation, okay? So you just average it out by month, January through December. So here's our dry season. The blue is water flow into the bay. And here's our rainy season. And then you look at the size of the algal blooms. It matches it pretty well. There's a little bit of a delay there, because they have to grow, but you see it matches pretty well. Well, we also have wet years and dry years because of things like El Nino and so on. So we look at long-term annual averages. Here's some wet years, and here's some dry years. And again, it tends to correspond pretty well. Well, at this point, I'm showing a connection between the sugarcane farms and these harmful algal blooms, which had caused huge environmental problems in Florida Bay and so on. And a lot of people weren't happy with that, not surprisingly. And so some people were hired to counteract this. And basically, they wrote a paper and claimed there was absolutely no correlation between runoff into Florida Bay and the size of the algal blooms. Well, do you see a correlation? Looks like it to me. okay? And so we came to exactly opposite conclusion. I see seemed to be a pretty good correlation. These other folks said there was absolutely no correlation. How could you? And it turns out we were both using the exact same data set. Okay, well, what's happening here? Well, it turns out neither of us used the complete data set. Okay, the complete data set included all of Florida Bay. I took the data at these 4 stations here where the bloom occurs. These other folks that opposed me, they took just the data in the blue area. They cherry-picked the data to take the blue area where there's no algal bloom. So of course you couldn't find a correlation between runoff and algal blooms. There was no algal bloom there, okay? Because you didn't have the phosphorus in the east. You only had the nitrogen. You have to have the nitrogen and the phosphorus, which is mixing there in the middle. So you have to be really careful. There's a lot of misleading statements out there. A lot of games that can be played. Okay, so anyway, I conclude that the phosphorus is natural. It's been there for millions of years. And interesting, well, let me back up. I said it's low here in the east because that calcium carbonate limestone sucks it up. Well, what would happen if you could somehow release that phosphorus from the limestone? Again, I'm not going to go into chemistry, but it turns out the Florida Department of Transportation did that experiment for us. This here is the road that goes from Homestead down to Key Largo. Okay, it used to be a 2-lane road, and they wanted to widen it. Well, it was lined with mangroves, so they chopped down all the mangroves, chopped them all up, and just dumped all the organic matter into the water on either side of the causeway there. Okay, that does several things, but anyway, it makes the sediments more acidic and more anaerobic, and that's exactly what you need to release the phosphorus from the limestone. And here's the result. This is before the bulldozing of the mangroves, and then here is when they bulldozed the mangroves, summer 2005. And all of a sudden you see this spike of phosphorus, and then you get an algal bloom. That's the green here. And of course, as the algae grow, then they pull the phosphorus back down. But that bloom lasted about 2 to 3 years. Okay, and again, that was a bloom of cyanobacteria. Again, we'll come back to that a little bit later on. So again, it just confirms what's going on here. And this again shows again here. Here's the causeway going from Homestead down to Key Largo. This is abundance of phosphorus before the bulldozing, and this is the phosphorus after the bulldozing. And here's abundance of algae before the bulldozing and after the bulldozing. Okay, well, you put this all together. Okay, this is from Nutrient Biobank. I have more data than what I'm showing here, but basically, essentially all of Biscayne Bay and eastern Florida Bay are essentially phosphorus limited. You get to the middle of the bay, it shifts over, and then the whole west coast is nitrogen limited because you've got a lot of phosphorus, naturally occurring phosphorus. Okay, so now we're going to look at implications here. Remember, we back pumped lots of nutrient water from the sugarcane fields into Lake Okeechobee. And then that water is pumped down the Caloosahatchee River to the west and the St. Lucie Canal to the east. I'll point out, the Caloosahatchee River was not initially connected to Lake Okeechobee. That was done by humans later on. Okay. Now, I've gotten funding to do research. I've done a lot of research on the west coast. I have not had funding to do research on the east coast, except for Biscayne Bay. Okay, so I'm going to focus, I know you're more interested in the east coast, of course, but I'm going to talk a little bit about the west coast. Okay, here's Lake Okeechobee, okay, and these red dots indicate high abundance of algae, specifically cyanobacteria down the river. Here's just a picture of these blooms of cyanobacteria on the Caloosahatchee River. Okay. At certain times, like after a hurricane, massive amounts of water comes pouring out of the Caloosahatchee River. You see all this dirty, here's the mouth of the Caloosahatchee River here. Here's Fort Myers there, and Sanibel Island. You see this massive amount of dirty water coming out of the river. It has a huge impact on the ecosystem. And every time I do a cruise along the west coast, I always see higher abundance of algae right there near the mouth of the Caloosahatchee River. If we take averages during the dry season, low abundance of algae in this region, Here's Sanibel Island again, okay? And then in the wet season, high abundance. Same seasonality that I showed you earlier for Florida Bay. Here's 5 years worth of data, Charlotte Harbor. The blue again is river flow coming down the river, and then the green is the size of the algal blooms in Charlotte Harbor. Pretty good correlation. Here's Pine Island Sound, same thing. Estero Bay, same thing. Okay, well, different years, because of different circumstances, you get different species of algae sort of being dominant and so on. Some years, it's this species here. It's a dinoflagellate called Carinia brevis. It's called the Florida red tide. And it's well known because it produces this toxin here called brevetoxin. Okay? So when you get large blooms of it, you get massive fish kills, harbor filled with dead, rotting fish, kills manatees, dolphins, sea turtles, and so on. Okay? Taking 50 years worth of data on the red tide, and the red and orange is a hot spot. So the basic hot spot for red tide is between Tampa Bay down to Naples, okay, and along the coastline, okay, less offshore. Now, every so often, the offshore current called the loop current picks up a little bit of this red tide, carries it all around the southern tip of Florida and up the east coast in the Gulf Stream. Maybe once a decade or something, you'll get a little bit of up here. But it doesn't like it up here, and so it basically dies out pretty quickly. It's not naturally occurring here on the East Coast. Okay? It's a big problem on the West Coast. So again, we take a lot of data to average it out. I mean, every year is different, but on average, again, from January through December, the blue is river flow. Here's your dry season. Here's your wet season. Dry season. Okay? And, oh, and I'm sorry, that's rainfall. The blue is rainfall. The orange is your river flow down the Clusaz River. And then the red is the incidence of red tide. So again, there's a delay because it's a slow-growing algae, but you do see that correlation. Here I've taken a transect from the shoreline going offshore. Okay, blue is salinity, the salty, and the salinity goes down as you move to the coastline because that's your freshwater runoff, diluting the salt. And then the red is your abundance, average abundance of red tide. It clearly goes much higher along the coastline. And again, that leads you to believe that it's the nutrients in that freshwater runoff that's causing this bloom. Same thing, same transect here, except the blue is what it looked like 60 years ago, and the red is what it looks like today. In other words, on average, you've got maybe 15 times more red tide today than you had 50 years ago. Okay? You just look at the overall abundance. The densest blooms you got 50, 60, well, 70 years ago now, I guess, is like 2.5 million cells per liter. Today, or recently, 35 million cells per liter. Clearly, a lot more red tide now. To get more red tide, you need more nutrients. And I can't think of any natural sources of nutrients that would have increased 15-fold in the meantime. Okay? So again, a lot of politicians and other people were getting very nervous about that and they've always said red tide's natural. Well, it is natural. But I argue human activities are making it worse. Okay? And so I'd publish this stuff, and so then some people were paid money to, or some consultants were paid to come up with a report to claim there's no connection between the increase in red tide and nutrient runoff. Well, interesting enough, you look down here in fine print, it says, in consultation with state scientists. Whereas I took the raw data and did my statistical analysis. These folks were told by the state to break up the data into 4 categories. Okay? And so anything over 100,000 cells per liter is category 4, which means we went from category 4 to category 4, which means no increase in red tide. It depends on how you manipulate the data. You have to really look at this stuff carefully. They're always playing these kind of games. Okay? Okay, so what's leading to the increase? Well, first of all, the blue area here is the natural watershed of the Klusiach River. Remember, it was not connected to Lake Okeechobee, okay? But of course, then what happened was the northern third of the Everglades were drained for agriculture. The huge Kissimmee River Basin was drained, largely for agriculture, ranching, and so on, okay? And what's happened in all these areas, you went from wetlands, which are natural sinks for nutrients, to activities which are sources for nutrients. So that's a huge increase in nutrients, plus the watershed has increased about fourfold. So not surprising, there's a lot more nutrients coming down the Klusiach River than you had, say, 50, 70 years ago. Okay. Again, this shows the history of the sugarcane farms, a lot more nutrients going back down the Klusiach River now. People, look at Collier County, 1950, 6,000 people. Year 2000, 250,000 people. Huge increase in number of people. Sewage, lawn fertilizer, all these things. Okay. And here, a colleague of mine took a core. What's nice about taking sediment cores is you can look at the history of an ecosystem. So here's a core from Charlotte Harbor, and you can use ISOOPS data. So this is the bottom of the core is dated about the year 1800, and the top of the core is the year 2000. So in all these nutrients here, what you see is this dramatic increase in nutrients in the last half of the 20th century, which corresponds to the huge increase in number of people and increase in agriculture. And also remnants of the algae. You can see that in the cores as well, dramatic increase the latter half of the 20th century. Okay, the effects. Okay, well, even before they knew what was causing these toxins, before they discovered that species of algae, it was called neurotoxic shellfish poisoning, because shellfish will eat these algae, but it's only vertebrate neurons that are affected by this neurotoxin, not animals like shellfish. So shellfish eat the algae and they're not affected, but they accumulate that toxin in them. So then if a human comes along and eats the shellfish, they get very sick. Okay, so it's called neurotoxic shellfish poisoning. Okay, that's not too common anymore because the state does a good job of monitoring for red tide, and they shut down harvesting of shellfish when red tide is present. Okay, but interesting enough, if you look at hospital records during times of red tide, there's about a 40% increase of people having gastrointestinal disorders. The main effect today, however, is that this toxin gets up in the air, like an aerosol, and it irritates. It's a neurotoxin, irritates the eyes, nose, throat, and lungs. It's like a tear gas. And again, you look at the hospital records, okay? It's about during times of red tide, there's about a 50% increase in people going to the hospital for various types of respiratory distress. Okay, now I want to shift back to the cyanobacteria. Most of those other blooms I told you about were cyanobacteria, okay? One in Florida Bay was, the one there in South Biscayne Bay here, that was. Okay, here's a satellite image of Lake Okeechobee. You see these massive blooms of cyanobacteria in the lake. That's the surface of it. Here's the Klusiach River, blooms of cyanobacteria. Probably, I don't know if you've seen that picture before, that's the St. Lucie Estuary, a number of years ago. We had cyanobacteria as well. And here's when they start dying. Some of you have seen this perhaps. And now you see why they get the name blue-green algae. They get the green from the chlorophyll, which is the photosynthetic pigment, but they also have another pigment called phycocyanin. It's blue in color. What happens when they die, they have this thick scum of cyanobacteria at the surface, and they're baking in the sun. As they die, the chlorophyll degrades faster than does the phycocyanin. So places that you've lost the chlorophyll, you still have that blue phycocyanin there. So those are the two pigments. That's how they get the name blue-green algae. And by the time they're dying, it looks like that. It smells really awful. Okay, well, one of the first toxins that was discovered, and probably one of the best known, produced by cyanobacteria, is microcystin. It was discovered like 100 years ago in Australia. A bunch of sheep died. Okay? And so trying to figure out what was going on. Well, turns out all these sheep were getting their drinking water from this pond over here, and they see this big bloom of cyanobacteria. So scientists get these algae, study them in the lab, and that's how they discovered this compound. There's a large complex molecule here called microcystin. Okay? And it's responsible for a lot of animal deaths, human deaths, and lower doses, gastrointestinal disorders, and so on. Okay? But these things happen fairly quickly, and so all the animals or people that are affected by this, they all happen at one location at one time. But by studying the biochemistry of this microcystin in the lab, you discover it can also lead to long-term liver damage. It's a tumor promoter, and it's now linked to liver and colorectal cancer. Well, these things take 10 or 20 years to develop. Suppose microcystin did not cause any immediate effects, and all those sheep or people only start dying at different times and locations 10 or 20 years later after drinking that water. How would you have ever linked it, 10 or 20 years later, how would you ever link it back to them drinking water from that one pond? You have no idea what was in that pond 10 or 20 years earlier. Okay? So this is one of the problems we have. Almost all the toxins we have discovered, produced by algae, is a result of people or animals getting sick all about the same time and location. It's a classic view, classic situation is a bunch of people get sick, end up in the hospital or whatever. What do they have in common? They all ate at a certain restaurant last night, and they all ate the clams. So you track it back, where those clams come from. Well, those clams are harvested from an estuary over here. You go look at that estuary, and there's a bloom of a certain species of algae. You now study that algae in the lab, and you discover a new toxin. Almost all of our toxins have been discovered that way. Okay? It's going to be very hard to discover a toxin that has no short-term effects, but has only long-term effects. By accident, however, we think we've discovered one. Okay? Oh, actually, let me back up. So anyway, you look at all these different kind of compounds that can be produced by algae. Some just smell bad, or can make the drinking water taste bad. Or if you go swimming in the water, it's like your skin is on fire. It's what we call a dermatoxin. Okay? Or it causes gastrointestinal disorders. And then in the longer term, neurological disorders, liver damage, neurodegenerative diseases like Alzheimer's, Parkinson's, and ALS, or cancer. Well, it's going to sound odd, but I call these the good toxins because you feel the effects right away, and you can do something about it. You know roughly what's causing it. You can take care of it. But what's much more insidious is if you're exposed to a toxin, it has no effect on you right now while you're exposed to it, and only 10, 20 years later, you come down with Alzheimer's or cancer or something. Okay? And this is the compound that we think has these properties called BMAA. Okay, and there's good evidence now it can lead to neurodegenerative diseases such as Alzheimer's, Parkinson's, and ALS. Now, these are often called the tangle disease because the characteristic is over time you get these proteins tangling up in your brain cells until it kills your brain cells. So just slowly killing off parts of your brain, okay? And what we know is they're mostly environmental. It's only like 5% genetic. I mean, there are some genes that can make you more or less susceptible, but it's primarily got to be environmental, and for many decades we've been trying to figure out what those environmental factors or triggers are, okay? The one exception is Huntington's disease. That is 100% genetic. We know the gene involved in that. Okay, but all these others, there's got to be some environmental factor. So we now hypothesize that BMA may be one of those factors. These are the types of protein tangles that you see inside your brain cells when you develop these neurodegenerative diseases. Well, it's a very interesting story. Okay, this goes back to World War II over in the Western Pacific. Okay, the Americans and the Japanese were fighting over the islands of the Western Pacific. And on the island of Guam, about half the people are the native Chamorro people. The other half are either Americans or other Asians who have migrated there for various reasons, okay? About 10 to 20 years after World War II, a large fraction of the native Chamorro people came down with mixtures, horrendous mixtures of Alzheimer's, Parkinson's, ALS. Okay? About 100-fold increase in these diseases. Okay? And for many decades, people couldn't figure out what was going on. Okay? And then a colleague of mine, Paul Cox, came along and took a very different approach. He's actually not a traditional medical researcher, he's an ethnobotanist. So he took a very different view. He actually went and lived with the people, observed what they did, and so on. And what it turns out is during World War II, there's warfare all around, they didn't have a very good food supply, okay? And they were hurting. But for the first time, they had access to guns. There's 2 species of bats that live on Guam, so they use these guns to shoot the bats. Before that, it was pretty hard to catch a bat, so they didn't eat very many. But once they had access to guns, okay, they started shooting all. In fact, they drove both species of bats to extinction, pretty much, on the island of Guam. Okay, so what you see here, here's the abundance of bats on Guam. It plummets during World War II, and then 10 or 20 years after they gorged themselves on all these bats, you see this 100-fold increase in these neurodegenerative diseases. Well, they drove the bats to extinction, so they didn't eat them anymore, and then the incidence of these diseases went back down 10 or 20 years later. Okay, well, what was happening here? Okay, I'm just going to go ahead and go here. What you find is, Paul Cox calls them flying foxes, but they're fruit bats, okay, and the Chamorro people are eating them, okay? And what we found was they had high concentrations of BMA in them. Remember this number, 3,500. Well, these fruit bats were eating the seeds of cycad trees. The cycad trees had symbiotic superficial cyanobacteria living on their superficial roots. And they were producing the BMAA. They're producing low concentration of BMA, and this was now this toxin is biomagnifying up the food chain, and so the flying foxes have really high concentrations of BMAA. Okay, so that was the hypothesis, and that's what's causing these neurodegenerative diseases among these people. It was interesting, the Americans who didn't eat fruit bats, or the other Asians who didn't eat the fruit bats, they did not come down with diseases. Only the native Chamorro people had this rather unique diet. So here's the data. Here's concentrations of BMA in the brains, and you see high concentration in the brains of the Chamorro people. Down here are, I know you can't see these numbers from there, but anyway, down here are, these are your controls. These are Canadians who died of heart disease or cancer or whatever. You do not see BMA in their brains. Okay, so that all makes sense. But then here are 2 Canadians who did die of Alzheimer's, and you find BMA in their brain. So it tells you it's not just this really unusual food chain that you had at one time in Guam. There's other ways of getting BMA into your brain. Presumably, these Canadians were not eating fruit bats from Guam. Okay. Well, a colleague of mine, Deborah Mash, she runs the brain bank at the University of Miami Medical School. So she looked at Americans who had died of ALS or Alzheimer's, and sure enough, you find high concentrations of BMA in their brains. Okay, in your controls, people died of heart disease or cancer or whatever. Let me see if I can get rid of that. Okay, yeah. Controls, you do not find BMA in their brains. And here's another control, Huntington's disease. Remember, I mean, you could argue what's cause and effect here. You could argue that having a neurodegenerative disease causes your brain to accumulate BMA. If you don't have neurodegenerative disease, you don't accumulate it. But here's your other control, Huntington's disease. It's a neurodegenerative disease, but it's genetic. And you do not see the BMA. So that tells you which way the cause and effect is. It's the BMA that's leading to the neurodegenerative disease. So when I saw her data, it was unpublished at the time, I'm thinking, here, I've been studying all these blooms of cyanobacteria in Florida. Okay, we need to see if this is a serious problem for Floridians here. Okay, so let's go back to Florida Bay. Okay, big bloom, that's cyanobacteria there. And a colleague of mine gave me some pink shrimp from that area. Here's one, 3,000, almost as high as those fruit bats in Guam. Here's another one about 1,500, half that of the fruit bats of Guam. You wouldn't want to eat shrimp from Florida Bay where that bloom is. Okay, remember this bloom here over southern Biscayne Bay where they had bulldozed the mangroves. Now this was not a good sample because a colleague of mine had done some research up in this region here, and it was toward the end of the bloom, and so I didn't have much problem. So I said, well, let's just take a look at those samples. He just had extra samples in his freezer. Say, well, let's just take a look and see. But again, animals move around and so on, so let's just see what happens. And a lot of variability, as we expected. But here is a blue crab, 7,000, twice as high as the fruit bats of Guam. Another one, 5,000. Here's a puffer fish, almost 7,000, twice as high as the fruit bats from Guam. Caloosahatchee River. You got these blooms of cyanobacteria in the Caloosahatchee River. You see people fishing there a lot of times. You had fairly high concentrations, ranging from about 500 to 2,500. So significant amounts of BMA in those fish. Okay, St. Lucie Canal. Well, like I said, I don't have much data over here. I haven't done much. But I wanted to go to the very top of the food chain. You do get nutrients and cyanobacteria from Lake Okeechobee down the St. Lucie Canal into the southern part of the Indian River Lagoon. And so what I want to do is go to the top of the food chain, and that is the dolphins. Okay, it's an endangered species, which means every time they find a dead dolphin, they do a full autopsy to find out what killed it. Okay, and they preserve the tissues and all that. It took me about a year to get the permit, but I was able to get a permit to get the brain tissue of some dolphins, 6 dolphins. Okay. Right, well, that just shows you the blooms you get over there. Okay. And sure enough, you see high concentration of BMA in their brains, similar to what you see of humans that have died of Alzheimer's and so on. Here's one, however, did not have BMA in its brain. The autopsy should have been hit by a boat. Okay. The other 5, the autopsies could not find any obvious cause of death. Okay. However, a colleague of mine now, David Davis, has been going into the details of this, biochemistry of the brains and so on, and he's now seen, in terms of the chemical signatures and so on, the same things that you see in human brains, humans who've died of these neurodegenerative diseases. So it does look like these dolphins are dying from this PMAA. Okay, go back to your high school biology. What's the mechanism here? There's several mechanisms, but one of them is BMAA looks very similar to serine, which is an amino acid we all need in our proteins. So what happens is you have a transfer RNA that's designed to, with a certain code, reads the DNA, and it's going to then use that to inject serine into the protein as you build up the protein. Well, the problem is this transfer RNA can sometimes get confused between serine and BMAA. Okay? So what you have to have for functioning proteins, you need to have the proper sequence of amino acids. Okay, so it folds up properly, so it has its proper biological functions. So we're going to use this like a slinky here. If you put BMA in there instead of the serine, that changes the folding of the protein, and you get this tangled mess. That's the source of the tangled proteins you see in the brains of people who come down with these neurodegenerative diseases. We think that is the mechanism there. Okay, and since then, lots of evidence along these lines. For example, you go to Sal Lagoon, where there's a large fishery for oysters and mussels, and people eat those, have a much higher incidence of ALS. And it's because they have large blooms of cyanobacteria in that Sal Lagoon. Wisconsin, blooms in Lake Michigan, and people who eat the fish from Lake Michigan also have a much higher chance of coming down with ALS. Okay, a colleague of mine up in New England's been doing the same thing, and others have been doing it in places in France. If you live near a lake where there's blooms of cyanobacteria routinely, you have a much greater chance of coming down with ALS. Other colleagues of mine have fed vervet monkeys with BMA And within 6 months, you see those protein tangles starting to develop in their brains. I worked with another colleague of mine at the medical school, Grace Lai. She works on the neurology of Drosophila. She uses Drosophila as a model animal for seeing what happens to nervous systems. And so what I did is, well, here, the black and blue lines are the controls. This is the normal lifespan of a Drosophila fruit fly, about 3 months, about 80, 90 days or so. Then what we did is we took some and we exposed, well, I got samples from Lake Okeechobee during a bloom, and we exposed the flies to it so they could smell, they could breathe the air, because this BMA can get into the air. And you see it shortens their lifespan considerably. And Grace also, again, looked at the details of the neurons, and so again, she could see how the BMA was screwing up their neurons in various ways as well. Okay, to finish up here then, let's go back for a second. Think of all the different pharmaceuticals that we use. Think about one of the earliest, like penicillin. Where do a lot of these drugs come from? A lot of them come either directly from plants and animals, what are called natural products, or we take those compounds and modify them a bit for certain functions. So the pharmaceutical industry is always looking for new antibiotics, new antiviral compounds, antifungal, and anti-cancer compounds. Okay, so they spend huge amounts of money just going all over the world looking for new compounds. They go to the rainforests, the coral reefs, all over the world, including cyanobacteria. They've surveyed lots of cyanobacteria looking for new compounds that might be useful as a pharmaceutical. And they found many hundreds of very unusual compounds with very complex chemical structures. First thing to note, these are not natural byproducts of normal metabolism. These compounds are produced for specific purposes. For the most part, we have no idea, but probably it's microbial warfare, okay, just like penicillin was. Large numbers of these compounds. So think about it. If you are exposed to if you're exposed to blooms of cyanobacteria, you're getting exposed to these compounds. We have no idea what their function and what they might do to you. So again, let's go back to that slide there. Some of these compounds will just smell bad, taste bad, or whatever, skin irritation. Those we'll discover. We've probably discovered a lot of those already. But what about the ones down here that will only have an effect on your health 10 or 20 years after you've been exposed? That's much more insidious because you have no idea you're being exposed, and it's going to cause major damage. You can't do anything about it, really. Okay. So the bottom line here, we're at 8 billion people now. We're continuing to add more and more people. That means needing more food, having to add more fertilizers. That leads to more sewage. That's inevitably going to lead to more algal blooms, and therefore we're going to be exposed to more and more of these algal toxins and these unknown compounds, of which I suspect some of those are going to be very harmful. And so let me stop there, and I'll take questions. Yes.
When you say that people that live on lakes which have the cyanobacteria blooms are exposed to these toxins, what's the path for the exposure? Is it eating fish from the lake?
Yeah, well, that's a good question. We don't really know for sure. People just...
Could you repeat the question?
Oh, yes, okay. So you see people who live near these lakes with cyanobacteria having a higher chance of coming down with ALS. What's the mechanism? It could be that they eat more fish from the lake than people that live further away. I don't know if that's true or not, but one's mechanism. And the other is the toxins getting into the air and they're breathing it. I might just mention, and some of you may know about this, I think it was like 1918, somebody had some really large blooms of cyanobacteria here in the St. Lucie estuary. And people who live on the canals or whatever, and same thing happened over in Fort Myers, same thing. A lot of people have their homes right next to a canal, and they put their boat there. Some people actually had to abandon their homes. The smell was so bad, and they were getting sick. And so it does indicate these toxins are getting into the air. We just haven't really nailed it down from a scientific point of view that we can actually prove it at this time. Yes?
Are you finding, is there any evidence that there's more neurological illnesses over on the west coast by Fort Myers than there is anywhere else in Florida?
Not that I know of. Oh, yeah. So is there any evidence that there's a greater incidence of these neurodegenerative diseases on the west coast than the east coast? Well, first of all, remember, the west coast, the major bloom over there is the red tide, which is producing brevetoxin, which is nowhere near as bad as these BMA or microcystin. The other thing is, well, I'll just briefly mention, I actually was going to work with some of the medical people to actually look at exactly that question, but we needed to get all the medical data, and the state blocked us. I mean, you have to, obviously, it's medical data, it has to be anonymous. You can't have people's names and all that, but there are proper ways of doing that, and I was working with the medical people who do this all the time. We could never get the data to look at exactly what you asked.
Larry, we have a question from one of our Zoom viewers. Does anything else make BMAA besides cyanobacteria?
There's some reports that some species of diatoms and dinoflagellates may as well. So again, we don't know the full story yet. Yes?
2 years ago, there was some research that said that the cyanobacteria toxin was being uptake in agricultural food products because of irrigation. Can you elaborate on that a little bit?
Yes. So the question is, yes, there are some papers, scientific papers, that have shown that food crops can pick up BMA as well. And what it is, you fertilize your crops, and if you're getting your water from the irrigation canal, so your nutrients will go into the waterways, and if you're now using those same waterways to irrigate your crops, okay, you're getting blooms of cyanobacteria in that water, and now the plants are taking it back up, and they're going to be picking up that BMA as well. Yes. So at least so a number of papers have shown that there is some amount of BMA.
How significant is that compared to animal products or shelf?
It doesn't seem to be as much. The problem is it's a biomagnification. And so if you have a long food chain, say from algae up to fish, it's a long food chain, so you get a lot more biomagnification, or up to, say, dolphins. But if it's going just to the plants, you don't have as much chance of getting biomagnification. But there's clearly some BMA in some of the plants, yes.
Let's grab another Zoom question. What's the half-life of these toxins? After an outbreak, how long does it take for them to dissipate from the environment?
I don't know. It's a good question, though. Oh, yeah, sorry. Yeah. So, yeah, I don't really know. I don't think I've read a study of anybody doing that, actually. Yes?
Given all the projects to reroute water to the Everglades and get it cleaner to go out, and not have as many flows coming out of the east and west, that should presumably cut down on all this, correct?
Yeah, the question is, we have this Everglades restoration project, okay, and the whole idea is go back to the original flow, and almost all of that flow is to the south, down into Florida Bay. Because right now, a lot of it's going to the east and west, down the Caloosahatchee River and St. Lucie, okay? If they succeed in doing that, yes, it'll make things better for you here, make things better for people on the west coast, make things worse for Florida Bay. Because remember, I showed you the data of every time you have more water flow into Florida Bay, you get larger blooms. But another aspect of that is that whole project, it's like a $20 billion, it started out as a $7.8 billion project, now it's up to like $20 billion. The whole project was aimed at taking the phosphorus out of the water. But I just showed you nitrogen's a problem. They got it backwards. I've testified about that, and they don't listen. So yeah, if they're successful in dumping more, it's better for you folks, but it's going to make things worse to the south. You saw the data. Every time you pump more water into Florida Bay, you get larger algal blooms. It's again one of the reasons why there's strong argument against this idea that people played games with the data. Yes.
So I know you're about to projection influences that are highlighting the algae, but what about the climate change?
Could you speak a little louder? I couldn't quite hear.
I can repeat it into the microphone. So he said a lot of your conversations have been about anthropogenic influences on algae blooms, but what about climate influences? Is that right, Nick? Okay.
What kind of?
Climate influences.
Oh, climate, okay. Yeah, you see a lot of people saying, well, people are sort of blaming everything on climate change now and higher temperatures. Higher temperature cannot generate more algal biomass. The only way you can get more algal biomass is more nutrients. So that's got to be the primary thing. Having said that, this is a question of which species of algae. And in general, cyanobacteria can tolerate higher temperatures than other species of algae. So you're more, so if you have a lot of nutrients, the question is, it going to be a non-toxic species or a toxic cyanobacteria? At higher temperatures, you're more likely to get a toxic cyanobacteria. So secondarily, temperature could be increasing these toxic blooms as well.
Let's grab another Zoom question. Is there a way to remove BMAA from your body after you consume it?
Some people are arguing that if you eat a lot of serine-rich, or just eat a really serine-rich diet. Remember, BMA competes with serine for that transfer RNA. And so it's really the ratio of those two. What you want to do is have a high serine to BMA ratio. I don't know if there's any evidence that it really works, but it's been hypothesized and they've been looking into that.
What kind of food is serine?
Some people are actually eating serine pills, pure serine. You can actually buy it in these stores and stuff.
Does serine occur in natural?
Oh, yeah. We all have serine in our bodies. It's one of the fundamental amino acids. So all living things have serine in them. But you're right. I mean, some foods have more serine than others. Just off the top of my head, I know sweet potatoes are particularly high in serine. But you'd have to look it up and find out. But yeah. I mean, my approach really is just to avoid these food sources that have been exposed to cyanobacterial blooms.
Oh, and that relates to a question that one of our Zoom attendees has asked. We're all being encouraged to eat more fish and seafood. How do we know whether it's safe to eat?
Good question, yes. Well, first of all, if you go offshore, out in the middle of the ocean, where you have tunas, mahi, things like that, you don't get harmful algal blooms out there. That's really low nutrient environments, and anthropogenic nutrients just don't make it all the way out there. So it's basically freshwater organisms in your coastal waters. That's the ones. You really don't want to eat the fish out there in the Everglades. There's really two reasons. One is the BMA, but the other is methylmercury is another issue. And that's where it gets complicated, because things like some of the large tunas and swordfish out there in the open ocean, they have high methylmercury. So, the fatty acids and everything is really good in the fish. So, some people tell you want to eat more fish, but then other people say, well, you don't want to eat more than one swordfish meal a month because of the high methylmercury. So, it's different problems with different types of food. But generally, offshore fish are going to be much better than things inshore.
This was a great presentation. Thank you so much. Super informative. I have a question, but to address the gentleman's question before, there's all these projects to improve our system, right? But I feel like a lot of the things we're doing are downstream, where we should be improving the upstream, right? The nutrient loading sources. So, but my question is, with the cyanobacteria, we see it when it shows up in blooms because it's fluorescent green and blue. What we're seeing now is what looks like some fairly clean water, like in our southern IRL right here. I do a lot of paddling there, and I've been witnessing some significant blooms. Last year, it was Lingia, or Dapis maybe is the appropriate. But this Lingia, I saw Lingia toxin as one of your compounds.
Right.
And then this year, like right now, currently, I think it's hookweed. I don't have the species genus name, but there's another huge bloom right now, and you don't see these things because they're under the water.
Exactly. Yeah, that's one of the problems is, oh yeah, so he's pointed out that there are times you don't see an algal bloom because what you see is this thing's floating at the surface and it's really dark green water or whatever, okay? And he's pointing out that's not always the case. And yeah, you're right. I mean, what's happening, a lot of times these algae don't just disappear. What they do is they sink down to the bottom. Because a lot of times there's more nutrients in those sediments. And so, yeah, just because you don't see green water at the surface, like you observed, which will give us a film of algae on the bottom. And it might be pretty clear water up at the surface, but they're down at the bottom. So, in fact, that's actually one of the things we found. When we go through our data, it tended to be the benthic-oriented animals that had higher BMA. So I think, like in Florida Bay, I did a study, and generally there's like 20 times more algal biomass sitting on the sediment surface than in the water column above. And so what we saw was like crabs, shrimp, and benthic-oriented fish, those are the ones that eat food off the bottom. They tend to have higher BMA than the ones that are up in the surface. We don't have enough data to really prove anything, but that's just what we happen to see with our data. So yeah, that's a really important observation that, you don't always see these things.
Any other questions here at the library? All right, let's finish with a question from our Zoom audience. What is the Florida government doing about the BMAA problem? Are they investing any money in research or finding any solutions?
Not that I know of. Oh, yeah. Not that I know of.
How do we know that was the answer? All right, folks, let's give Larry a big thank you.